All right, chapter 3, last part. This is on cell division mostly, and it's pretty long, so I'm going to try to go as fast as I can. All right, so the cells have to divide when they get too big, or when they they either have to divide or die when they get to that point. And so there's two parts to the cell cycle. Most of the time, healthy cells are in interphase. And then when they're dividing, they're in mitosis, which is cell division. Interphase is when the cell is just growing and doing its job, like it's just being an eye cell or whatever. And then sometimes the cells will divide. There are some cells that never divide, like your red blood cells. They just die. Nerve cells also rarely divide. So there's different phases that we're going to look at that you've hopefully seen before. But in interphase, the nuclear material is uncondensed, and it's being used, so it's being coded. It's in a relaxed state so that it can be accessed, like the DNA polymerase can access it, or RNA polymerase, and code it. And then when it's in mitosis, it has to get organized and condensed. So these are the subphases of interphase. This is the main phase that cells spend most of their time in. There's gap 1, where the cells are growing and doing metabolism and just doing their thing. Some cells stay in gap 1 forever and never divide, and so that's called gap 0. But if a cell is going to divide, the next step would be synthesis, and this is where it copies its DNA. And then gap 2 is where it prepares for division. It copies organelles, gets everything, like takes inventory, and makes sure that it's ready. So here's your cell cycle. The green, this is all interphase. You've got gap 1, synthesis, and gap 2. And then mitosis, when the cell's dividing, is right here. So out of a 24-hour period, a cell would probably easily spend like 20 hours in interphase and only 4 hours doing mitosis, if that. Most of the time, cells are in interphase. So in interphase, if it goes into synthesis, the DNA is going to be replicated. It's going to be copied. This happens by the DNA strand unwinding, unzipping, and then there's a replication fork or bubble, and that's where the DNA is being replicated. And you have little RNA acting as a primer, and the DNA is being copied. The one that does that is DNA polymerase. That's an enzyme because it ends in A's, and it attaches to the primer, and then it brings in new nucleotides that match, that follow the A to T, C to G rule. There's 2 strands. One of them is called the leading strand, and it goes continuously. The other one is called the lagging strand, and it goes backwards in pieces. And then DNA ligase is an enzyme that binds them back together. So finally, at the end of synthesis and interphase, you're going to have 2 daughter cells, but 2 daughter strands of DNA. But each strand of DNA is going to have half original DNA and half new DNA. And that's what semi-conservative replication means. So here is our DNA polymerase. This is your DNA strand. This was your original back here. Let me get my pointer out just to make sure you can see it. So right here is the original strand, and it's being unwinded here. And this kind of relaxes it. And then your DNA polymerase here is going to put in new pieces of DNA. You also have a primer, an RNA primer that comes in and primes it. But this is your lagging strand, and it goes away from the replication fork, and then your leading strand goes towards the replication fork. So you can watch that video, and it's helpful to see it in real life, like actually moving. Okay, so then if a cell does go into mitosis, because of maybe it's a damaged cell, you get your finger cut, you break your arm, or maybe it's because you're growing, or they're just replacing old cells just for maintenance, you go into mitosis. Some cells never do this, like nerve cells don't do it very often, cardiac, skeletal, not super often, but skin cells do it a lot, right? So mitosis goes in 2 parts. There's the mitosis part, and then there's the cytokinesis. Cytokinesis is when the cell actually separates into 2. Mitosis is basically the nucleus dividing. And it has to be controlled really, really well. If it doesn't, that can result in things like cancer or other problems. So here's the cell cycle. Here's mitosis. You've got the nucleus dividing here, and then cytokinesis. It can start way back in anaphase, but cytokinesis is when the actual cell divides. So mitosis, nucleus divides, cytokinesis, the cell divides. So mitosis has 4 phases: prophase, metaphase, anaphase, telophase. So you should know those. The acronym you can use is PMAT. That's the order. So I'll show you what happens in each phase. And it's pretty logical, like how this happens. But remember, this is division of the nucleus, not the cell. Dividing the cell is cytokinesis. So so prophase has 2 parts. There's early prophase and late prophase. And then sometimes they'll have like in between phases, like pro-metaphase. So in early prophase, the chromatin is going to condense into chromosomes. And these are really organized, and it makes it easier to divide the cell. And then you're going to get centrosomes to appear. And those are made of microtubules of cytoskeleton. They're going to help guide the chromosomes and separate them to the right place. And then you have another structure called a centromere, and that's going to connect the 2 replicated sister chromatids together. So you started with 1, and then during synthesis they replicated and you have 2. And the centromere is a little belt that joins them together. Late prophase, the nucleus dissolves because it has to get out of the way, and then you're going to have microtubules attaching, and they're actually going to attach to the poles of the cell, on the equator is what they call it, so that they can pull the chromosomes apart opposite directions. So here is prophase. And this is a cool view of it. This is a fluorescent microscope. But you can see the centrosomes forming. They're going to move to the poles. Nuclear envelope is dissolving. Your chromatin is condensing. I think this is actually more, this is more interphase. This is what it looks like before. So then, early prophase, the centrioles start to move to the ends. These are called asters here. And then this is a mitotic spindle, but it's all made of microtubules. The chromosomes are condensing, and then this is late prophase. Chromosomes are condensed. The spindle fibers are attaching to the centromeres, and you can see the chromosomes are condensed, and the nucleus is pretty much gone. It's out of the way so that the DNA can divide. Okay, then you have metaphase. Metaphase means middle. This is when the chromosomes line up in the middle because the mitotic spindles pulled them there. So here's your mitotic spindles pulling them to that midline, and all those chromosomes are there. And I think it kind of looks like a little spider that got squashed. Then anaphase is a very quick phase. You almost miss it. That's when the chromosomes are pulled apart to opposite ends of the pole of the cell. So here they are. They're getting pulled apart, and then eventually they're going to make it to opposite sides. That's what it looks like. So in telophase, that's the last part. That's where the chromosomes are on opposite ends. The nucleus starts to reform. The chromosomes relax, and the spindle fibers disappear, and everything kind of goes back to normal, except you're going to have 2 main nuclear spots where the nucleus is going to be in each cell. And then cytokinesis is when the cell separates. It starts like in late anaphase or early telophase, so it's kind of happening at the same time, and the 2 daughter cells are pinched apart. So here's telophase moving to the poles, nucleus is reforming, chromosomes are relaxing, and then the cell is pinching apart in cytokinesis. Okay, so there's a lot of things that control the cell doing this right, because there's a lot that can go wrong, and if it goes wrong, that can lead to cancer. So your cells have, I call it like the gas and the brakes of cell division, so the go and the stop. So they have certain things that are go signals that say yes, you can divide. So one of them is critical surface to volume ratio. That's when the cell gets too big and it's going to have to divide or it'll die. So divide or die. The other one is chemicals. So sometimes there are hormones that the cell is going to get as a message. Remember how cells communicate with each other? And it's going to say hey, it's time to grow. So those 2 things are going to cause cells to divide. The stop signals in healthy cells are when there's not enough space. So when cells get close to each other and start pushing on each other, they have this thing called contact inhibition, and that tells them hey, there's too many cells here, let's just stop and rest and do our jobs. There's different proteins that do this work that help the cell move from cycle to cycle exactly when it's supposed to. Those are called cyclins and CDKs, and they work together. So it's kind of like having the cop and the cop car, and they're the ones that control whether the car, like traffic, can go or whether it stops. Okay. So cyclins accumulate during interphase, and then CDKs are cyclin-dependent kinases, and the cyclins bind to them. And then if they're bound together, and there's different ones, so it has to be the right one, that's going to let the cell go into division. After it goes to division, the cyclins go away. The CDKs are always there. So the CDKs are like the cop cars. Cop cars are always parked in the parking lot. The cyclins cycle in and out, go up and down, depending on what the cell's doing. And if there is a cyclin and a CDK, a cop and a cop car together, then they're going to allow the cell to go through division. So this is done at checkpoints, is what we call it. So there's different points in the cell cycle when the cell checks to make sure everything's okay. And so one of them happens in gap 1, and it's a really important checkpoint. It makes sure that everything's okay and that the cell is ready to divide. And if it doesn't go through that, it's gonna go into gap 1, or it could even kill the cell. It could do a self-destruct and kill the cell if everything isn't correct. So gap 1 is right here. This has been in the other pictures. Gap 1 checkpoint. And then there's also a gap 2 mitosis checkpoint, and there are other ones. They're just showing you 2. Okay, so the purpose of DNA is to code for proteins. So it has the information needed, like the files to code for proteins, which is like printing a file. Like I think of the file, and then you have the printer. We have DNA and proteins. The way that the code is programmed is it has those A's, G's, T's, and C's that we've learned about. And it's coded in 3 bases at a time. So like you would have these 3 nucleotides, these 3 bases, and that's going to code for the amino acid proline. There's 20 different amino acids. This is one of them. Or GCC will code for arginine. So each 3 of these letters, every combination of letters codes for 1 amino acid. Some letters code for more than 1 amino acid. In genes, you also have exons and introns. So you have some parts of the DNA code that don't actually code for proteins. They're kind of considered junk DNA, but they're probably not actually junk. They probably actually have important roles, and they're finding more and more about that. But exons are the parts that are used, that are coded, and then introns are the parts that are not used. So the introns, before the RNA strand leaves the nucleus, the introns are removed and cut out, and then the exons are used and come out of the nucleus to make protein. Okay, so RNA is the molecule that leaves the nucleus and brings the information to the ribosomes, and that's because if DNA left, it would be destroyed. RNA does get destroyed when it leaves the nucleus, but first it'll code for proteins. There's some differences too that we've learned about. RNA has a uracil instead of a thymine, and RNA is single-stranded, and it has a ribose sugar instead of a deoxyribose. And then we've heard of these 3 types of RNA: messenger, ribosomal, and transfer. They all have a role in making protein. So messenger RNA is that little messenger that sends the information from the nucleus. It's only going to code 1 gene at a time. So it's like a flash drive, putting 1 file on a flash drive and then taking it out of the nucleus. And the flash drive is going to be destroyed, but first you'll be able to print the file and then destroy the flash drive. So when DNA is changed into RNA, it's called transcription. That's the process, and it happens in the nucleus. And another type of RNA is the ribosomal RNA, and that's what ribosomes are made out of. And this is made in the nucleolus. And then tRNA works together with the ribosomes to make protein. So it's coding the mRNA at the ribosome, and then it drops off the amino acids. It carries them in and drops off the correct amino acid. And that process is called translation. Okay, so these are the 2 steps to protein synthesis. Transcription is DNA to RNA. That happens in the nucleus. Translation is RNA to proteins. That happens on the ribosome. So here's transcription, and then here is translation. This mRNA processing part is where the introns are removed. So you're only left with exons here, and then it leaves the nucleus and gets coded. Okay, there's some different things that also will control whether transcription happens, and those are called transcription factors. So this will control whether the mRNA is even made. One way that it does this is by loosening the histones. So those are those little proteins that the DNA coils up on, like little hair rollers. And if it's too tightly coiled, then the DNA can't be coded. So the histones control that. And then there's also like a sequence of DNA before the gene that you're trying to code. And it's kind of like a switch. And so if that switch isn't turned on, then the gene will not be coded. So this is a way to turn on and off genes. Sometimes it does it at the right time, sometimes not. And then you also have RNA polymerase, which is the one that makes the RNA, and it has to be able to bond to the right spot. So those are 3 ways that it can control which proteins are being made. There's 3 steps to transcription. One is called initiation. So remember, transcription is the RNA being made in the nucleus. So the RNA polymerase is going to separate the strands. It looks a lot like DNA replication. Then the RNA polymerase comes in and brings in the right nucleotides that match the DNA template. And then termination is when it stops and the RNA falls off, and then it goes to get processed. So this is initiation. RNA polymerase grabs it. Elongation, it starts to make the mRNA, and then termination, it's done. There's your piece, and it's going to go get processed and used to make protein. So processing of RNA, a couple things happen in the processing stage. One thing that happens is the introns are removed. There's these special proteins called spliceosomes. It removes the introns, leaves the exons to get coded. So that's one thing that happens. Oh, and it doesn't say the other things. The other things are that you put a cap and a tail on the ends of the mRNA to protect it. Then where are we going? We're still in transcription. So this change from DNA into mRNA takes a few different parts, and then later you're going to change it into protein, and that has other parts. And then we already looked at this, but the genetic code is based on A's, T's, C's, and G's in DNA, or AUCG in RNA. And you have 3 of those together, so it could be any combo, AUC, AUG, CUG, GCU, UAC, any of those. There's actually 64 different combinations. And those 3 letters together, those are codons, okay? So 64 different combinations, and they only code for 20 different amino acids. And so there's a lot of codons that code for the same amino acid. 3 of them are stop codons. They don't code for anything. They just stop the process. And then there's one that's a start codon, but that does code for an amino acid. So here's what I mean by some of them code for the same thing. Let's look at like serine right here. Serine is an amino acid. It's coded for by 4 different, at least 4, actually 6 different codons. Let me show you. So all of these code for serine, and these 2 code for serine. Some of them, there's only 1 codon that codes for them, like methionine. That is the start codon, and only AUG codes for it. Leucine has 6. Some of these, like I know these are weird words, but some of them might be familiar. For example, lysine, you could actually buy at the drugstore. It's a supplement. So you're just buying that amino acid. It's supposed to help with cold sores. So, not tyrosine, where's tryptophan? No, where is it? Why can't I find it? Tryptophan here is is in turkey, and that's what they say makes you sleepy after you eat turkey, but I don't think it's really true. And then these 3 are the stop codons. Okay, so the role of tRNA here. tRNA is that other type of RNA. Remember, there's lots of other kinds, but this is a really important one. This is a molecule that brings in the matching amino acid to the ribosome, and it matches to the mRNA. And that's how protein is coded. So this end right here is called an anticodon, and then this end is called an amino acid. And it comes in and binds with the mRNA and drops off the amino acid. I already said that. I'll try to find a picture of it here. The ribosomes is where this happens. So the ribosomes are like the mediator between the mRNA and the tRNA, and that's where all of this happens and where the protein is made. There's 3 steps to translation: initiation, elongation, and termination. So those are your 3 main steps, and they all have different enzymes and things. These are the names are the same as the 3 steps for transcription. So initiation is when the ribosome connects to the mRNA, and it connects in this certain spot. There's the P site and the A site and the E site, and it starts in the P site in the specific spot, and then the tRNA is going to join it. So that's your first step. It has to happen at a methionine codon because that's the start codon, so that's your AUG. Then elongation happens, and the ribosome is going to move down the mRNA, and as it goes, then the tRNAs are going to come in and bind to it and drop off amino acids, and so it's going to shift down these 3 sites. The ribosome is going to move down the mRNA just like, and then the tRNA is coming in and binding. So then, yeah, once the A site's open, a new one comes in, and it's just like a little factory. It's just bringing in amino acids and binding them. This isn't as detailed as I wanted, but so we have the ribosomes here. The blue is the mRNA, and then the little yellow things, those are the amino acids. They become a polypeptide. So the little ribosome is just chugging. I think we're going this way. Chugging down the little mRNA and making the protein that matches it. So just think of a little factory. Termination is the last stop. So once it hits a stop codon, that's where it's going to stop, and it's not going to code for another amino acid. It's just going to stop, and then the mRNA will be released. It'll go degrade. It falls apart, and then you have your protein, and it's going to go get processed by the Golgi. Okay, this is what I wanted. So here's your ribosome. I know this is really confusing, and it probably didn't help that I was just talking and not showing you. So here's your ribosome, and you have your mRNA, and the ribosome is moving down the mRNA, and these tRNAs right here, they come in and bind. So right here's your start codon, AUG, and they bring in the matching correct amino acid, which is methionine. And they're going to drop that off in a second once it can stick to another one, and the tRNA is going to leave, but it's going to leave the amino acid there, and then the tRNA takes off. So here is showing a tRNA coming in, and then here's showing a tRNA leaving, but it's dropping off an amino acid. And so they just keep coming in, switching places, and then eventually here's your stop codon. The polypeptide, which is made up of a bunch of amino acids, is now released, and you're not going to make an amino acid with the stop codon. It's just going to stop. Then it lets go of the mRNA, and then the mRNA is going to be degraded. The protein is going to be folded up into something. Okay, so we have this segment called the ER signal sequence on a polypeptide, and that's where the ribosome is going to connect to the ER surface. So that's going to help the protein be made better, and then eventually the protein will go off and be processed in the Golgi. Okay, so here is, what do we got? Here's the ribosome, here's the mRNA. It's making the protein, and then on the ER you have that little signal here where it's going to signal recognition particle, and that's going to start folding up the protein here and like getting it folded and processed, and then it's gonna leave. It's gonna butt off the ER and go to the Golgi, and this is your vesicle. So that's just a little like pre-processing step that happens to the protein. Okay, so I think we're almost done with this part. So you have DNA triplets, your 3 letters, your A, T, C's or G's and 3 letters. That's your mRNA codon. That codes for 1 amino acid. It happens at the ribosome. Okay, so here's your DNA, gets coded into, oh, here's your DNA, sorry. Then it gets coded into RNA. The reason I know this is RNA is because it has U's instead of T's. And they match, they're following the base rules, like A's bond to T's, but in this case, the A is going to bond to the U because it's a U on mRNA. Then an anticodon is going to match it at the ribosome and then drop off a matching amino acid. Okay, here's some other types of RNA. So I told you there were other kinds. One kind is a microRNA. That is kind of like it's a little monkey wrench, you know what I mean? Like it comes in and it's going to block the mRNA so that it can't be transcribed. So that basically silences the mRNA, or so it can't be translated. Ribose switches are little folded pieces of RNA that are little switches. They act as switches and can turn things on or off. And then you have siRNAs. They're kind of like the microRNAs, but they come from viruses. So they're finding more and more different weird things that RNAs can do that are really interesting that we don't know about. Okay, so when things don't go right, your cells have a mechanism called autophagy, where they can basically eat themselves and self-destruct. They become degraded. So the lysosomes are the ones doing that. They kind of let the enzymes out of the lysosome and they just digest themselves. You also have this ubiquitin protein. If there's ever a messed up protein, they can be marked for digestion. And then these proteasomes, another little some structure, will come in and chew them up. Also, there's a process called programmed cell death. This is where a cell will self-destruct. So this is what hopefully happens to all cancer cells. If the body recognizes that they're cancer, it'll activate the self-destruct sequence and break them down. But sometimes that doesn't happen, and that's how cancer happens. Okay, so when you started out from one cell. You were a zygote. You were made from a sperm and egg, and you had a bunch of files of DNA of genes. All of your cells came from that first cell. So all your cells have the same DNA, except for sex cells, like sperm and egg. They only have half the DNA. But you get chemical signals to your cells, and they tell your cells to turn on or off certain genes. So that's called cell differentiation. When your cells start to look like different types of cells, even though they have the same genes, and it's just because they're accessing different files, different genes. Okay, so when you're born, your organs are there, but they need to keep dividing as you get bigger. And you need to do mitosis if you have wounds. So there's 2 different ways this can happen. When you have accelerated growth, that's called hyperplasia, but sometimes it happens when it shouldn't. And then you can also have atrophy, which is the opposite, when your cells start to like break down. So think of like atrophy of your muscles. Your muscles lose mass and aren't working as well. And sometimes both of those are good. Sometimes they're both bad. So they just have to happen at the correct time. Over time, your cells age too, but they're not really sure like exactly how it happens. So there's 3 kind of ideas. One is the wear and tear theory. So like chemicals, free radicals break them down. One's the mitochondrial theory of aging. Free radicals are in the mitochondria are making them not run as well. And then autoimmune disorders might also be causing cell aging. Also, we have these little caps on our chromosomes called telomeres. They're like, you know, the little things on your shoelaces. They're like those. They protect your chromosomes, but over time they start to degrade. They get shorter and shorter, just like your shoelaces, and eventually they can start to break down your actual chromosomes, like the important part. Um, Telomerase lengthens them, but they're finding that it's not as common in adult cells. And so, oh, and sometimes cancer cells have too much telomerase and makes the telomeres too long. And so that's a problem too. So, okay. Let's see. This is a weird disease that they're showing. I'm almost done. Progeria. It's a disease that mimics aging. So like little kids look like they're aging quickly. So that's one thing they can study to understand aging. So it makes this defective progerin protein, and kids start showing it when they're like age 2. They start getting brittle bones, thinning hair, arthritis. They usually only live till about 20, and it shows up around age 2. And they're looking at drugs to help clear out the progerin. And then here's a picture of a child with that disorder. And that's it.